Okay, everyone, good afternoon. Today we'll be starting actually the material for exam number 5. Exam number 5 is going to be all on the cardiovascular system, and it's going to include chapter on the blood, a chapter on the heart, and a chapter on blood vessels. So we've got 3 lectures before that exam. That exam will be a week from Thursday. We have today to discuss the blood, we have Thursday to discuss the heart, and we have next Tuesday to remind ourselves about arteries and veins. Now tonight will be the last group of people who are having the lab on arteries and veins. And so the nice thing about this next test, I think, is that you have already seen much of this material. You've already been introduced to it in lab, you've already been quizzed on it in lab, or will be before you take this next exam. And so as I go through the blood, and as I go through the heart, and as I go through the arteries versus veins story, much of this will seem, at least, it won't be brand new, right? It'll be a review of some things you've learned, and there will be some new information added, but you already have a good chunk of it already working through your head as you think about this. That'll be exam number 5. Remember, I do have an error in the syllabus. My vocabulary slide numbers are incorrect, and we're going to pick up from wherever exam 4 ended and go through 92. And last time I went through some of those, 73 right, through 92. So picking up then, last time we went through the first 4 or 5, picking up with 78 on rami. Now, most of these terms I'm going to go with you today right now, we've already seen. So rami, ramus, remember that was a branch of the nerve that we saw coming off the spinal cord. And means back. To rehabilitate is to bring someone back into their normal habits, if you will. So to rehabilitate, to bring them back to normal function. Rect, we know, means straight, and I apologize for my typo on there. It should be IS at the end of rectus abdominis. But you know rectus means straight, rectum, straight. And reno, kidney. We'll be seeing a lot of renal terms starting next week in lab. That'll be lab number 10 will be over the urinary system and on the respiratory systems. We'll see a lot of renal structures, and then the renal system, the kidneys will be part of your final exam, as will the respiratory and the reproductive systems. Continuing on, reti. You've seen the retina. You know the retina is a network of photoreceptors. You've seen the endoplasmic reticulum. That was a network of membrane structures within the cell. You've seen reticular fibers, again, network-like fibers. And soon in the male reproductive system, you'll see the rete testis, which is a series of tubes, a network of tubes within the testes. So we've seen a lot of terms that have reti as a part of them. Retro, backward or behind. When we get to the urinary system, we'll see that the kidneys are described as being retroperitoneal. They are behind the peritoneum. They're behind the serous membrane of the abdominal pelvic cavity. When you look into your fetal pigs next time, and you've taken out the digestive organs, you will see the kidneys. They'll be there, but they'll be behind this layer, the serous membrane. So you'll have to go through that layer to actually touch the kidneys. Rhino, the nose, or rhinoceros, right? Big old nose. And rigor, stiffness. You've seen rigor mortis, the stiffness following death. Rage, excessive or abnormal flow. Hemorrhage, right? An abnormal flow of hemo, blood. Or diarrhea or menorrhea, rhea meaning a flow. So diarrhea, flow through the body, no fun. Or menorrhea, excessive or profuse menstrual flow. Rube or rubro, both referring to red. Now within the middle of this term, bilirubin, you see the ruby, the R-U-B, right, the rube. And bilirubin is actually what helps make your, part of what makes your urine yellow, what makes your feces brown. But this is actually a breakdown product of hemoglobin. Remember, hemoglobin is also red, what makes your blood red. So the heme portion breaks down and we get these bilirubin molecules. So rube means red, just think ruby. And then rugo, fold, in the inside of the stomach, the rugae, were the folds. And if you look at the word corrugated, you'll see RUG in there as well. Cardboard, right? Corrugated cardboard has that wrinkled kind of appearance on the inside. Sarco, flesh or muscle. We know it as muscle, right? But when you're eating the muscle of an animal, you're eating, it's flesh, right? And sarcolemma was the outer layer of a muscle cell. Sarcoma would be a tumor of a muscle. Sclera, hard. We saw the sclera, the outer hard layer of the eye. Scleroderma is a condition of an autoimmune condition where the organs and skin gets kind of tight or hard. And atherosclerosis, you see sclero in there as well, the hardening of the arteries. Scopo, to see, microscope, seeing small things. Endoscope, to see within, endo meaning within. Seb, grease. Sebum is the greasy stuff that comes out of your facial pores. Remember we talked about sebaceous glands, they were your oil secreting glands. And secto, to cut, to dissect, to cut. And I'll go through one more with you today. Semi, half, or partial. Now the semilunar valves in the heart, it means half a moon, right? Semilunar. Honestly, I don't see half a moon. When I look at the semilunar valves in any drawing that I've ever seen, it does not look like a half a moon to me. But that's where they got their name, and we're stuck with it. Sept, your septic tank is rather contaminated, right? And septicemia, right, would be contamination of your emia, of your blood. So emia, blood, septic, contamination, some sort of bacterial infection of your blood. Serrate, saw-edged, serrated edge. There's a muscle in the chest called the serratus anterior. We didn't have it this semester on our list, but it kind of from across the room looks like a sawtooth edge to it, the serratus anterior. And then finally for today, SIA, meaning a condition or process. And I don't know where I got that example. It doesn't even have SIA in it. So I apologize. The example there is not valid. But that gives us through 82. And we're going to go through 92. So we got 5 more to do each day. 5 on Thursday, 5 next Tuesday. Please, please, please don't wait until the weekend to get that going. Go ahead and get your vocab cards going. I think you will agree that many of those terms are now more familiar to you than they would have been 3 months ago, 2 months ago. That many of those terms have a meaning to something we've already seen, so you're not starting off fresh with this vocab. Okay, so here we go. Blood. And this is, of course, Chapter 17. I need to get my numbers updated for this new book. Now, Chapter 17 is on the blood. Chapter 18 will be the heart. Chapter 19 will be arteries and veins. Like I said, the good news is you've already seen many of these exact slides in lab, or something like it if Mr. Mueller presented a few different things to you. But you have seen this. You have heard this story. So kick back, relax, enjoy the review, and then wake up when I get to something new. Okay? Now, you know that blood is a connective tissue. And by blood, by connective tissue, right, all connective tissues are such because they have what? 1, loosely packed cells. You got red cells and white cells bouncing off each other, surrounded by a ground substance. In this case, the ground substance is a liquid called plasma. And there are proteins dissolved in that plasma, and we'll describe those proteins today. So blood, although it's a different kind of connective tissue, does meet the definition of a connective tissue. It is thicker than water. Duh, we know that. Temperature-wise, your blood's about 1 degree Celsius warmer than your body temperature. That makes blood a warm, nutrient-rich place for bacteria to hang out. So should bacteria get into your blood, they love it there. And so they'll kind of go nuts and they'll create a blood infection. Diabetics are more prone to blood infections because their blood has more sugar in it. So it has more nutrients for the bacteria. Now you can take your blood and divide it out into sections. And we'll do this in Biology 106. For this semester, we'll just describe it to you. But we'll take some blood, we'll put it into a thin tube, we'll then spin it in a centrifuge for about 5 minutes, and then we will see how the blood separates out. And when we do that, we will see that blood can be broken into 3 major layers. The bottom layer will be all of the red blood cells, the erythrocytes. It will, by average, account for about 44% of the blood volume. That is a number that represents males and females and all people on the earth, around 44% average. There will be a middle layer when you spin the blood down. It'll be a very thin layer. It may not even be visible as a layer. It may just be an interface between the bigger layers. This layer is called the buffy coat. And it's here in the buffy coat that one would find the leukocytes, the white blood cells, and the platelets. Finally, the top layer, and these are kind of upside down, the top layer will be all the liquidy plasma. It'll account for the other 55 or so % of the blood volume. And as we'll see, everything that's dissolved in the blood would be found in or dissolved within the plasma. So it'll look something like this. Now this looks like a big honking test tube with a lot of blood in it. What we'll use in the lab will be more microcentrifuge tubes, very thin tubes. But regardless, one would take some blood. And let me quiz you on this. It's been a while. What part of the body do we normally take blood from? The inside of your elbow is called the antecubital region, or the AC region. And what sort of person normally draws blood, is kind of specially trained to draw blood? Phlebotomist. And we know from our vocab that phleb means vein. We normally draw blood from a vein, and we'll talk more about those differences in lecture next week. You'll hear a little bit about arteries and veins in lab tonight, or you already have. Now, we take that blood. This is going to be typically venous blood, and there's good reasons why we take venous blood rather than arterial blood. The most obvious reason, anybody know? Why do we take venous blood over arterial blood when just drawing blood? Less pressure, right? If you hit an artery, it's going to squirt. It's going to be a lot more difficult to get it to stop bleeding. And the other thing is that arteries are more protected. They're deeper in the body. And for good reason, right? If you cut an artery, you're going to bleed more substantially. It's going to be more difficult to stop the bleed. The blood clotting will be more difficult. If you cut into a vein, the pressure is very low. The blood will bloop, bloop, bloop out. It'll clot very easily. And the veins are more toward the surface, right? You look at your wrist, and what do you see? I see blue. I see veins. Arteries are deeper. So you're going to have to go deeper if you're going to get into an artery. And most phlebotomists, they're in the business of just drawing normal old venous blood from the antecubital region. There are times where one draws arterial blood for gases, but that's not your typical, right? When you go to the doctor, just give blood, you're giving venous blood. What color is that blood? When you look at the blood bank and you look in the bag that they're collecting, or you look in the tube, in that vac tube, and they're drawing blood into an oxygen poor environment, that blood stays darkish, reddish, brownish, kind of bricky brown, doesn't it? That is your venous blood. Now, we say it's blue blood. It's not really blue, but we perceive it as being blue when you look at your veins, right? We see that bluish color. It's not really blue blood, but that coloring is what has influenced our decision to put blue as venous blood on models. Right? If you instead cut yourself and the blood was exposed to the atmosphere, that blood would be a much brighter red. Right? That's going to be oxygenated blood. Okay, so lots of information there just to think about. Now, you take that blood, you put it into a centrifuge tube, you spin it for about 5 minutes, and when you're done with all that, you will see 3 layers. The bottom layer, and I'll get to these information in a minute, that's going to be all red blood cells, and on average would account for about 44% of the volume of the blood. We would say that that is that person's hematocrit value. We'll talk about the hematocrit in a moment. But remember that hemato, blood, crit, means to separate, right? Crino was to secrete. Remember this from the C's. Crino was to secrete, endocrine secrete, and crit was to separate. So hematocrit, you have separated the blood into layers, and we're determining that the percent of red blood cells is clinically referred to as your hematocrit value. Significance coming. The little middle layer is the buffy coat, and then the majority of it is the plasma. Now let's dive in just a little bit more. I want you not to memorize these numbers as much to appreciate them. So on the bottom, all red blood cells. Look at this number. 4 to 6 million red blood cells per cubic millimeter. Can you all picture a cubic millimeter? Make a little cube with a millimeter on each side. That's a very small little box, isn't it? And in that box, you would be able to put 1 microliter of blood. So 1 cubic millimeter, you'll see it written like this, cubic millimeter, is the same as a microliter. Very small drop of blood, 1/1,000,000th of a liter. In that little drop of blood, there will be 4 to 6 million red blood cells. The very middle layer right here, the buffy coat, that is going to contain your platelets and all of your white blood cells, of which there are 5 different main types, and I'll introduce those to you in a few minutes. And then the plasma is the liquidy portion. That's going to be about 92% water. So your bloods are roughly half water, right? Roughly. And also in your plasma will be proteins. And we'll go through these different types of proteins in a few minutes. And remember that because it's water, it's a polar molecule. Many things dissolve in your blood quite well. So being carried through your plasma are things like sodium, potassium, calcium, all of the electrolytes, plus nutrients, glucose and waste products, and everything else is being transported through your blood. Also your gases. You have dissolved oxygen and dissolved CO2 traveling through your plasma, and the waste products as well. So we're going to break this down even further. Here's a term that you may not know yet. If I take your erythrocytes, if I take your erythrocytes and I add to it the components of your buffy coat, what's the buffy coat again? White blood cells and your platelets, right? So your platelets and your white blood cells make up the buffy coat. We're going to call all of that your formed elements. Right, so your red cells, your white cells, and your platelets collectively are referred to as your formed elements. The reason is they're not all really cells. And so you'll hear people say, oh, your blood cells. And I get it, but that's not quite accurate. Remember that platelets are not cells at all. They are little fragments of cells. And you may remember back in in lecture maybe 1 or 2, when we described the characteristics of life, remember in that hierarchy of atom to atom, I said that cells are the functional living thing. Right? And red blood cells really aren't living. Red blood cells don't have a nucleus. They don't have DNA. They can't copy themselves. They don't have much of a metabolism. So some people would even argue perhaps that red blood cells aren't really cells because they, quote, aren't living because they can't reproduce themselves. So we're going to use the word formed elements to describe all of these things in the blood. Now, if you go down and give blood to the Red Cross, you are giving whole blood. That's everything. That's your formed elements and the plasma. If instead you go down and you donate plasma at the plasma center, then you are giving away just the liquidy portion, and they're going to give you back your formed elements. They're going to give you back your red cells, your white cells, and your platelets. You can give plasma twice a week because what they're taking is pretty much water with the proteins, and your body can replace those proteins very readily. Whereas if you give whole blood, you can only give about every 6 weeks. It takes your body quite a while to recover from giving half a liter of blood. You only have about 5 or so liters of blood. So when you give blood, you're giving about 1/10 of your blood away, and your body's going to take some while to replace all those cells. And if you go into the blood bank to give blood and you're anemic, they're not going to let you donate because you're already lacking in red blood cells in the first place. So the last thing they're going to do is take even more cells away from you. Now, there are about 5 or so functions of blood. What does blood do for you? It's amazing, really, how many different jobs this stuff has. Number 1, pretty obvious, transportation. As your heart pumps, it's pushing blood. We get that, but what's being transported? Not only is it pushing around oxygen, and picking up CO2, but it's also pushing around nutrients and hormones and waste products, right? So everything that's being transported is being transported as it's carried through by your blood. Number two, and we talked about this long ago when we were in the integumentary system, the blood is helping to regulate your body temperature. You know that when you're hot, when you're exercising, your body is going to appear more ruddy or red in color. Blood will be flowing more toward the surface of your dermis and your skin, and your body is desperately trying to get rid of heat, right? Lower the body temperature. When it's cold outside, from now till next May, darn it, you will be more pale in color because that blood will move away from the surface, trying to conserve heat, and your vessels in the dermis will vasoconstrict. Vas means vessel, constrict to close down. So your vessels will vasoconstrict, and you will lose facial color, but you'll be conserving heat. Number three, we talked about pH back in lab 2 or 3. And reminder, pH is a scale from 0 to 14. 7 is considered neutral, like pure water. Things that are above 7 are alkaline. Things below 7 are acidic. Now, your blood pH comes in at about 7.4, and there isn't much wiggle room here. 7.35 to 7.45 is the normal range for your blood pH. Remember, this is a logarithmic scale. A change of 1 is a 10-fold difference in acidity. Your body can only tolerate a swing of half plus or minus. So below 6.9 and above 7.9, you're dead, right? The body cannot handle having a low or a higher pH by more than half a pH value on either side. So it's highly regulated, and the components of your blood, as we'll get into the kidneys and the respiratory system a little bit this semester and a much more next semester, we will better appreciate how the blood works with those systems to maintain your blood pH at about 7.4. Also, in lab tonight or last Thursday, you were introduced very, very briefly to the lymphatic system. And all you learned is that there is fluid exchange going between your plasma, your blood fluid, and your lymph, the interstitial fluid. And there's constant flowing in and out of this lymphatic fluid and your plasma. That fluid balance is very important for maintaining your normal blood pressure and your normal functioning. If, for example, you have too much fluid in your blood, that would increase the pressure and you would have high blood pressure. If instead too much fluid left your blood pressure would go down, but now you might complain about edema, fluid accumulation somewhere else, maybe in your ankles, or maybe accumulation of fluid around your heart or your lungs. So there's a very careful balance of fluid, and I'll come back to that a little bit more when we talk about the proteins within the blood. Just remember, solutes suck. Remember that phrase from long ago. And so we'll talk about how your blood pressure is maintained and how these fluid levels are maintained. And then finally, protection. You've got white blood cells traveling through your blood. Those white blood cells are an important part of your immune system and your ability to fight off infection. When we talk about blood typing, antibodies are mentioned, and you've been told that antibodies are produced to fight off foreign antigens, foreign things. But what kind of cells make those antibodies? Some of these white blood cells. And then finally, don't forget about platelets. Platelets are also protecting you against bleeding out, so they're also quite protective. And we'll talk about platelets as we finish up today. So lots of functions of blood. Any questions on what blood is doing for you? If you're having blood issues, you might go see what kind of specialist? Hematologist, right? A doctor who deals with blood disorders of all sorts is a hematologist. And you can see that clearly there are lots of systems that interact with your blood. when we get to this next test, and you have the obligatory questions about how does the cardiovascular system interact with other systems, remember blood is part of this. And already we've seen that blood interacts with your lymphatic system to keep your fluid levels balanced. We've seen that blood is made in your red bone marrow, in your skeletal system. We know that blood is going to send oxygen to your brain, to your liver, to your stomach, to your reproductive organs, you name it, right? And blood is going there. There is absolutely no valid reason on the planet Earth to leave those questions blank. That I can't think of anything that the heart or the cardiovascular system does in interacting with other body systems. Please don't make me beat my steering wheel all the way home. I do. My car is out of alignment. I think because I beat my steering wheel somewhere around the odoriferous Coopersville exit. Okay, not only does the smell get to me, but those blanks get to me. It's like, come on, folks, fill it in. And then I'll have someone say, well, I couldn't think of anything. I didn't study that. Okay. Okay, I rant. I'm sorry. Okay, let's talk about plasma. Plasma, the liquidy portion. Do you agree that plasma is water, right? It's proteins and it's other stuff. If I take the proteins out of plasma, I now call it serum. You may have heard of that term, serum. So serum is plasma minus the proteins. Now, the easiest way to get rid of those proteins, sorry, got a little disconnect here. So if you just take your plasma and you remove the proteins, that's called serum. Now, one way to do this is if you draw blood into a regular tube, very shortly thereafter, the blood will clot, get gelatinous. And if you were to remove that blood clot out, you now have serum. Because as the blood clots, all the proteins are being pretty much removed. So you take the blood, the plasma is a liquid, take away the proteins, now you have serum. The rest of it is largely water. Right? So serum is largely water. But when you go down and you give plasma at the plasma center, what you're giving them are clotting factors and some other things that are in very, very small concentrations in your blood, and they gather it from many individuals, and then they sell it for big bucks to people with hemophilia and other clotting disorders and blood disorders. Okay, plasma proteins. So I already told you that there are proteins in your blood. They come in 4 different groups, and they account for about 7% of your plasma. Let's just say 7%, that's a range of really like 6 to 8%. Now remember that percent is 1 of 100, right? Percent. So another way of thinking about that, if I had 100 grams or 100 grams of blood, 6 to 8 grams of that would be protein, and a gram of water is about a milliliter of water. So anyway, crazy little conversions there. But if you had 6 or 8 grams of stuff on a scale, it's not insignificant. That's a lot of stuff dissolved in every 100 milliliters of blood. Now these proteins come, like I said, in 4 groups: albumins, globulins, fibrinogen, and a category just called regulatory proteins. I'm going to tell you a little bit about each of these proteins, 1 slide each. Albumins, these are in the order of their abundance. Albumins are the most abundant of all of your blood proteins, and by size, they're the smallest. They make up a large percentage of your blood proteins. What I want you to think about for albumin this semester is that albumin is regulating your movement of water in and out of your blood. Remember that phrase, solutes suck. If you don't have enough albumin in your blood, what would happen to your blood pressure? Think about it. If you don't have enough protein in your blood, then what would happen to your blood pressure? If I don't have enough protein in my blood, that means water would not be drawn in, right? Solutes suck. Albumins are the major solute. They're going to draw water into the blood and raise your blood pressure. Now, what if you don't have enough protein in your blood? Then water won't be sucked in, right? Water will stay where? Out in your tissues. This explains, in part, those children we see from third world countries. They don't have enough protein in their diet. They got skinny little arms, very little muscle, but they have big bellies. The big bellies is fluid accumulation. What's going on? They don't have enough protein in their diet to keep their albumin levels high enough. So water is not drawn into their blood, and instead it stays out in their gut, and they get these big bellies. And people say, well, they're not starving, they're fat. No, they're not. All right, this is a product of insufficient nutrition, and they don't have enough protein in their diet. Make sense a little bit? Okay. So when you hear the word albumins, think fluid movement and regulation of blood pressure and fluid in the body. Now, you know albumins in a different way. Egg whites, right? Albumins, same thing. Egg whites, same thing in the chicken. Now, albumins have other roles, but we're going to focus only on the fluid regulation for this semester. The second group of proteins, most abundant group, are called the globulins. Now, that may be a new word for you, but you have perhaps heard of the immunoglobulins and more commonly the antibodies. So the globulins really are your antibodies. They are the second most abundant group of proteins in your blood. They are the molecules that fight off foreign antigens, aren't they? So they're a pretty big deal, and they're a big part of your blood. Number 3. Oh, and what kind of cells make these antibodies? B cells or B lymphocytes, absolutely. And those are a type of white blood cell. So your lymphocytes, the B cells that make the antibodies, are one of your white blood cell types. The third most abundant protein is called fibrinogen. Now I'm going to write a note here. Fibrinogen. Now, fibrinogen, it's a big old word, right? Is a protein that floats around in your blood, and it is soluble. That means it's dissolved, right, in your blood. However, the moment there's any sort of damage to your blood vessels, there are going to be enzymes released that are going to cut this fibrinogen protein into a smaller protein called fibrin. Now, fibrin is insoluble. So it's going to come out of solution and start causing or forming the blood clot. So fibrinogen is what's floating around in your blood, right? That's what's up in your blood, fibrinogen. But when your vessels get damaged, fibrinogen is modified to a protein called fibrin. And fibrin is going to make the clot, the fibrous clot of your blood. Now, one more word here. When you are clotting your blood, that process, and we'll dive into the steps of that next semester, but the process of stopping blood flow is called hemostasis. Now look at that word and don't be confused. Not homeostasis, right? That's the whole story on keeping things equal. Hemostasis, blood. Stasis, static, keeping things relatively the same. Stopping blood flow. Hemostasis. The last group of proteins is everything else. And this is a group of proteins called the regulatory proteins. It is going to include the enzymes, the clotting factors. Nothing here is in very large concentration, but collectively, again, this is what the blood center is all excited about when you donate plasma, because they're collecting these very relatively rare molecules accumulating them from hundreds of patients and using them to treat people with blood disorders. So those are the different proteins in your blood. And let me remind you about this term, solutes. Do you agree that the protein in your blood is a solute? Right? Solutes is everything dissolved in the blood. The water is the solvent. The stuff dissolved in that water is the solute. And together we make this solution called plasma. Do you agree that plasma is an extracellular fluid? Think about that for a second. Plasma is outside of your red cells, outside of your white cells. It's the stuff that the cells are floating in. So it is an extracellular fluid, right? Make sense? It's outside of the cells. Now, plasma is pretty much identical to interstitial fluid. What's the other name for interstitial fluid? Lymph, right? Lymphatic fluid. Lymph and interstitial fluid, same thing. Plasma and lymph are pretty much the same thing. They have pretty much the same concentration of nutrients, of waste products, of sodium and potassium. What's dramatically different between plasma and lymph is the concentration of oxygen. Let's talk about this for a second. How, when you dream about oxygen coming into your airways and entering your body, how does oxygen get to your tissues? Comes into the lungs. We'll talk about this next week with the respiratory system, or 2 weeks. That oxygen is going to jump onto hemoglobin. That's going to be carried on your red blood cells, right, and go to your tissues. In the alveoli, right, so we're going to grab that oxygen from the atmosphere and we're going to drop it over into the blood, and that blood's now going to be carried to your tissues. So now you're down in the tissues, you're down in some capillaries where the oxygen is going to jump off from your blood and enter into your tissues. What causes the oxygen to move, to go into the body, from the blood into the body? Diffusion. Now that's why we had to learn all this stuff a while back. Diffusion was, remember, the movement of molecules moving from a higher concentration to a lower concentration passively. No ATP, no energies involved. So that means that in order for oxygen to move from the blood to the tissues, the plasma must have more oxygen in it, and the tissues must have less. So oxygen moves out of the blood into the tissues. At the same time that that's happening, what's going from the tissues into the blood? CO2. So the reverse must be true. So what I'm saying to you, big picture, and we'll come back to it, is that plasma and lymph are pretty much the same, nutrient and everything else, except for their concentration of gases. Oxygen higher in the plasma, CO2 higher in the interstitial fluids, and that's why this gas exchange happens. And we'll break that down in greater detail, but I wanted to introduce that idea to you now. So again, there it is in print. The difference in concentration assures that oxygen will diffuse out of the blood into the tissues. Conversely, the difference in concentration of CO2 will assure that we're going to get rid of the CO2 from the tissues and put it into the blood for delivery back to the lungs and release from the body. Now here's one of those tables that I put up sometimes. It has nothing new. Anything that's extra in here that I didn't already put in the PowerPoints, do not worry about. But this table just reminds us that blood is, if we break it down, right? Or sorry, this is plasma. Plasma is 92% water, 7% proteins, and that includes albumins, globulins, fibrinogen, and the regulatory proteins. And then everything else would be the electrolytes like the sodium, the potassium, the nutrients, the fatty acids, the amino acids, all that, cholesterol, gases, and things like lactic acid and everything else that's being broken out. All the stuff that's floating in your blood is dissolved in your plasma. So that's the story about the stuff in your plasma, and that is the story about the functions of blood. The rest of our time, we're going to spend a few more minutes looking specifically at different formed elements. We're going to look at some details about red blood cells, then some details about white blood cells, and finally we'll finish up at the end talking about platelets. So erythrocytes. Okay, first let's remind ourselves what the formed elements are. It includes the erythrocytes. These are by far your most abundant. They make up more than 99% of all of your red blood cells. Their job is to transport gases, primarily oxygen, and also CO2 is riding on hemoglobin. White blood cells, their job, all about protection, immune system. Different kinds of white blood cells have different special jobs. Some white cells are better at attacking bacteria. Some white cells are better at attacking worms and parasites. Some white cells are better at attacking a virus. And others are phagocytic. They chew up things. So there's different kinds of white blood cells. They each have their own job. And then we talked about some of these also make antibodies. So lots of different jobs. We'll focus on many of those details next semester, but they're all involved with your immune system. Now, number-wise, they're relatively rare. They only make up 0.01%, very small percent of your overall formed elements. And that's because there are just so many red blood cells. Millions and millions, right, per drop. So far fewer of the white blood cells. Finally, there are platelets. Platelets make up about 1% or so of your formed elements, and they are all about blood clotting. So you have three distinct different types of formed elements with three quite different functions. There are some numbers here that I do want you to be responsible for. They'll show up in other places as well, but this table puts it all in one place. First of all, let's look at size. Red blood cells, 7.5 microns, or 7.5 micrometers. That's their diameter. Okay. When you are looking at tissues under the microscope, histology, and when your eye becomes trained and you are able to spot a red blood cell, it's like having its own little ruler in there, because that little red blood cell is telling you that it's about 7.5 microns in diameter, and it kind of gives you a little internal ruler to gauge the size of everything else in your field of view. What do these red blood cells do? We know that they're transporting oxygen and CO2. They have a lifespan of about 120 days, okay, about 4 months. They're made, they float around for 4 months, they carry oxygen. The numbers, again, millions per microliter. You're going to see different numbers floating around because men have more red blood cells on average than women, and then altitude can have an effect. So if you were to talk to a nurse or a doctor in Denver, they would tell you, well, the average hematocrit here, and wherever they tell you, is going to be higher than what it is here in Muskegon or more closer to sea level, because hematocrit changes, and we'll talk about that in a minute again, but altitude can have quite an impact on the percent of red blood cells. But again, don't memorize the numbers, but again, millions per drop of blood. White blood cells, 1.5 to 3 times bigger. So that means they're going to be closer to 11 or 12 microns, up to as big as around 22 or so microns. They're bigger. Their job, white blood cells, their job is to help with your immune response. Their lifespan varies from hours to almost your entire life. These are the cells that have memory. These are the cells for which you get vaccinated. You receive a vaccine, your immune system, some of your white blood cells remember that vaccine and will keep you covered, right? Keep you, quote, immune or protected against that challenge for the rest of your life, or at least for many, many years. So some of these cells are with you forever, most of your life. Others of these are short-lived, depending upon what's going on. And their numbers, again, 5 to 10,000. I'll have that number written on a slide coming up. but relatively rare compared to the millions of red blood cells. Then your platelets, they're small, less than a quarter of the size, about 1 to 2 microns of a red blood cell. Their job is all about blood clotting. They're going to float around for about 8 to 10 days. So they're rather short-lived, just 8 to 10 days, about a week or 10 days. And their numbers, although pretty high, hundreds of thousands, are still dwarfed by the number of red blood cells. So what I'm going to do now is just go through each of the different formed elements. We'll start with red blood cells. We'll talk about those for a little bit. Then I'll move on to white blood cells, and we'll finish up with a few details about platelets. So talking about red blood cells, again, if you separate out your blood, spin it down in a centrifuge, look at the result, you will see those 3 bands. The bottom band will be all red blood cells. And that bottom band is going to account for males, somewhere around 42 to 56% of the blood volume. That is considered the individual's hematocrit value. The more red blood cells you have per volume, the higher your hematocrit. Guys are going to be hovering around 50%, plus or minus 50%. Okay, that's how I remember it, right about half. Okay? Whereas women are going to have, on average, a lower hematocrit, high 30s to mid 40s. Okay, so lower 40s on average. So that means a woman who has a hematocrit lower than 38% would be considered anemic. She has insufficient blood cells at that moment. Now, what would her symptoms be? Tired, right? You're anemic, you're tired, you don't have good color, right? Your color's not going to be good, you're going to have less rudiness, less red. You're going to be tired, you don't have the ability to carry as much oxygen to your tissues, so you're going to be very tired. Now, a guy would have to have something less than 42% to be considered anemic. Children, the numbers vary. We won't worry about those today. And then altitude also can have quite an effect on hematocrit. So if you fly today out to Denver and you try to do your normal routine, exercise or walking around routine, you're going to find yourself very winded, very tired. And the reason is, what's different in Denver? Yeah, the common thing is, oh, there's less oxygen. It turns out there's still 21% oxygen in the atmosphere in Denver. The difference is there's less atmospheric pressure, right? And that's what really matters here. That's why when you're looking at cooking instructions on the Betty Crocker box, it tells you at high altitudes, you got to change the cooking instructions because altitude also has an effect on cooking, okay? But high altitude has less atmospheric pressure. And basically what that means is that as you expand your lungs and your lungs start filling up with air, what pushes the air into your lungs is the pressure in the atmosphere. Where there's less of that pressure up in Denver than there is here. That means that less oxygen will be pushed into your lungs per breath. Well, your body says, I'm not getting enough oxygen. So in response to that, it starts making more red blood cells. So your body will kick up the production of red blood cells to normalize that lower oxygen atmospheric pressure. And as a result, your hematocrit value would rise. Okay? And it takes about 3 to 5 days for that to happen. So you're going to feel kind of crappy in Denver for the first 3 or 5 days. After that, your body will normalize, and you'll be going about your normal routine without any issue. Now, if you want to have an advantage, if you're running a marathon, and you want to have an advantage over the other folks, spend a couple weeks up in Denver. Right? Train up there, get your body to naturally have a higher hematocrit, fly back down to sea level, and within the next couple days run the race. And you will already have a higher oxygen carrying capacity than your competitors because you've been training in the high altitudes. It's what our athletes, right, the fine athletes do that. All the Olympic training centers are in Colorado Springs or Lake Placid. They're all at high altitude places. One, because they have snow there for the Winter Olympics, but also they have that high altitude training experience. And, so if you want to win the marathon, go train up there. It's also why, as crews are doing, as people are climbing up K2 or Kilimanjaro or Everest, right? They can't just go up the mountain. They have to set up base camp, and then they have to travel only so far. And then they rest for a couple of days, and then they go further. They're not just resting because they're tired. They're resting and waiting for their body to adjust to the higher altitude, which in part is their body kicking up their production of red blood cells. Okay, because if you just zip up the mountain, even if you can physically do it, you'll be huffing and puffing because of the altitude changes. Okay, so a few more details about red blood cells. I've already told you that mature red blood cells do not have a nucleus. That means they don't have DNA. That means they don't have the ability of dividing and undergoing mitosis, and they can't make more of themselves. Their job is to transport oxygen, and what's so cool is because they don't have a nucleus, they don't have much of a metabolism. That means that they're carrying around oxygen without using it. Right? It's like the most efficient transport system you could ever imagine. So the very cell that's carrying the oxygen really isn't using up much of it. Any other cell in your body will be sucking up that oxygen and using it for its own metabolism. So it's a really efficient, wonderful system for carrying oxygen around your body. We've seen that biconcave shape. Remember what that means? You look at a red blood cell from the side, and it has that dog bone look. It has those two indentations. That's what the biconcave means. And you know that they're kind of flexible because we saw back in lab 2 or 3 where in different environments red blood cells can crenate or they can swell. So you already know they're kind of flexible. They can bend and kind of elbow their way through small blood vessels. They can even line up in single file when going through little capillaries and little blood vessels. They hang around for about 120 days. They can't divide. They don't have a repair mechanism. So they can't fix themselves and they can't make more of themselves. So they have a limited lifespan, about 120 days. And what happens is that your spleen and your liver, they're getting a lot of blood. One of the jobs of the spleen, one of the jobs of the liver is to basically be watching for damaged red blood cells. And it will begin to have them broken down. So your spleen and liver break down your red blood cells as they're getting destroyed. There are macrophages there. They're going to chew them up, destroy them. Well, if they live for 120 days, think, that's kind of close to 100 days. Therefore, about 1%, right, a little bit less, 1% of all of your red blood cells are being destroyed every day by your liver and your spleen. They're getting worn out. That means, too, that every day your body's having to make 1% of all of your red blood cells. And that's millions and millions and millions of cells per day. I think the numbers are somewhere around 25 million cells. And I don't want to misquote here, but it's still astronomical, either per second or per minute, right, that you're making in your body. Millions of cells every second, easily, in your body are being made. And many of those are red blood cells. So here's that little picture, right? Red blood cell, there's that number, 7.5 microns in diameter. If you look down from a superior view, look down on a red blood cell, you have that little rafty looking image. It would look the same from the inferior view. And when we look at this little blood smear, we see all these red blood cells, all biconcave in their shape. Now, what is it that carries the oxygen? I've been saying over and over that red blood cells carry oxygen. The actual molecule that carries that oxygen is a molecule called hemoglobin. Heme, there's blood again, right? Hemato. And there are 280 million of these protein molecules in every one of your red blood cells. That's a pretty astronomical number. So that means that every drop of your blood has 4 to 6 million red blood cells. And each of those red blood cells itself has about 280 million molecules of hemoglobin. Millions times millions. It's like our national debt every second, right? Just numbers we can't even appreciate. Huge, huge numbers. Now you know that when hemoglobin is well oxygenated, that it burns bright red. Right? Your oxygenated blood, the blood leaving the lungs, is bright red. Blood, however, that is less than fully oxygenated appears more deep reddish browny color, and we perceive that blood as blue through our skin. I'm going to skip over, no, I'm going to skip over a slide in a moment. I'm going to go through this one first. I'm going to skip a slide, show you a picture, and then come back. The hemoglobin itself, this is all brand new. The hemoglobin itself is a protein made up of 4 subunits, 4 parts. Each of those parts is referred to as a globin. Now don't get confused there. Not a globulin. The globulins, the immunoglobulins, the antibodies, that's different. This is a globin. Think globe, round, spherical. So this is a portion of the protein called a globin. There turns out there are 4 of them. 2 of them are called alpha, 2 of them are called beta. In the center of each of these globins is a non-protein portion called a heme group. Now you know why they call it hemoglobin, right? Heme in the center of each of the globins. And it's the heme group includes an iron ion, Fe, iron. And it's to that iron that the oxygen binds. So every hemoglobin has 4 globins, every hemoglobin has 4 hemes, and therefore every hemoglobin can carry a maximum of 4 oxygens. Another disconnect moment here. Let me get this back. So I'm going to skip over a slide and then come back to it and show you a picture of hemoglobin. Here's a cartoon representation. So the 2 units on the top are alpha, or sorry, beta on this picture. The 2 yellows are the beta. So imagine what you have here are 4 subunits, right? They all together, that makes up 1 hemoglobin molecule. 2 betas, 2 alphas. In the very center, what looks like Saturn, that's the heme group, and the red center represents the iron ion. Okay, so you've got 4 heme group on every hemoglobin. You've got 4 oxygens that can bind to that. And when all 4 oxygens are bound, then hemoglobin is a pigment. It's a colored molecule. It shines bright red. And when there's less than all 4, then it becomes a little more dull, right? More of a dark red color. Going back, then, just a couple more details. What I just said to you, every hemoglobin can carry a maximum of 4 oxygen molecules. Now that binding of oxygen is described as being weak, W-E-A-K. Right? It's not very strong. And that makes sense if you can take a little magic school bus ride with me and imagine that you are traveling on a red blood cell and you're traveling down to the tissues and you've got an oxygen molecule next to you. You want that oxygen to be able to quickly jump off, if you will, into the tissues. If the oxygen was strongly bound to the hemoglobin, it wouldn't let go easily, and it wouldn't jump off in time. So this is a very weak association between the hemoglobin and the actual oxygen. As we talked about before, right? The gas exchange is all about diffusion. It's all about concentration differences, and we wouldn't want the hemoglobin to be grabbing onto the oxygen very tightly. Now, back in chapter 3 or 4, we spent some time talking about what cells do, like transcription and translation. And remember that whole story, the DNA to the RNA to the protein. And one of the mutations that I described to you at the end was for sickle cell anemia. That mutation, remember that mutation, that 1A to T or whatever it was, I forget, switch causes the protein to change and make the blood cells strangely shaped. Well, that protein is hemoglobin. Okay, so going back and pulling that all together, it's the hemoglobin that has the mutation in it. And that mutation can cause the red blood cells to change their shape into these sickle-shaped cells. what actually happens, remember I told you there are 280 million molecules of hemoglobin inside the red blood cell. That mutation causes those hemoglobin molecules to polymerize, to make a chain. And they stick to each other and make a long chain. You already know that red blood cells are squishy. They can change their shape rather easily. So inside, the hemoglobin molecules have made a long chain, if you will. Now, they're not this big, of course, but they're making a long chain. They're sticking to each other, and that's going to change the shape of the cell into the sickled shape. That polymerization, that chain reaction occurs when the hemoglobin is exposed to high altitude. So people with sickle cell are told not to go to the mountains, do not go flying, do not go to high altitudes, and also minimize stress, whatever that means. Because stress and altitude tend to trigger this sickling event. Now, in a moment, actually next time, I'll be telling you more about arteries and veins and capillaries. But remember, this guy is about 7.5 microns across, normal red blood cell. I'm going to tell you later on that capillaries are only about 8 to 10 microns across. So a red blood cell is already rather claustrophobic. It's already a very tight squeeze for red blood cells to go through a capillary and deliver the oxygen. And I'll tell you right now, this red blood cell, this sickled red blood cell, is going to clog up any little capillary. So now you've got these sickled cell red blood cells blocking oxygen flow through your capillaries, through your vessels, and causing tissue damage and potentially even death if you're blocking the blood flow to the brain or to the lung.
So if they were to have a blood transfusion?
That's what they do. That's what they do. So when they go in, when they're having a sickling attack, and it's very painful because they're having tissue damage wherever those blood clots are, they'll give them blood transfusions and try to, quote, dilute out these, they'll draw blood out and give them new blood, right, transfusions. And they'll try to get as many of these sickled cells out of their circulation. And they'll also give them blood thinners, and they'll give them, Coumadin and other things to help push these blood clots out. So they'll give them TPA, the same kind of stuff they'll give a heart attack patient or a stroke patient trying to thin their blood and break up the blood clots. They'll give these patients some of those kinds of agents. They'll give them transfusions, basically to get rid of those bad cells and minimize the clots. Hopefully do it fast enough to minimize long-term damage. But what happens is they just keep having these attacks, and that damage is cumulative. So over time, their kidneys, their liver, their other organs start getting really beat up by these repeated attacks. Okay, that's all I'm going to, one more slide on red blood cells, and then I'll ask for any questions. This is just the life cycle of a red blood cell. So what do red blood cells do? They are made where? In your bone marrow, right? So we're looking at spongy bone, up in the bone marrow. And they're going to travel through your circulation for about 120 days. As they get tired and worn out, they're going to be broken down by the spleen and the liver. They're going to recycle this. And what's going to happen is that the iron will be recycled. The heme group will actually become part of a breakdown process that gives your stools their brown color and gives your urine some of its yellow color. So the heme part will get broken down, and that's recycled or broken down, and then some of the other cellular components will go back and be reused to make even more red blood cells. So it's a big recycling cycle going on. What's important, all blood cells made in the bone marrow, travel for 120 days, broken down by the liver and the spleen. So that's all I'm going to discuss about red blood cells. I'm going to start moving into white blood cells, but before I do that, any questions or concerns, anything I've discussed so far about blood or red blood cells? Yes, ma'am.
The red blood cell, you said that the sickle is caused from lack of protein?
No. The sickling is hemoglobin, the protein that's inside, that normally carries oxygen. That same protein, hemoglobin, has a mutation. That mutation, and I didn't say this will be helpful. That mutation does two things. It causes the hemoglobin molecules to make a long chain under stress, and that changes the shape of the cell into that sickle shape. The other thing, though, is that the hemoglobin no longer carries oxygen as well as it should, and so the patient is more anemic. So that's where those two terms are coming from. Sickle cell anemia. Sickle cell because of the shape of the cell that can change. Anemia because the mutant hemoglobin doesn't carry oxygen as efficiently as normal hemoglobin does. Okay. So there's two parts to that disease. Even when they're not having sickling moments, they still are anemic. They don't have a lot of energy. They don't have the same oxygen capacity. They're probably not going to be running a marathon or doing any excessive exercising because they are already anemic from the disease. Does that clarify what you were asking? Okay. Yes?
You talked about pressure changes when flying. What about the opposite, like scuba diving?
Scuba diving, same thing. Yep. So people with any sort of sickle cell disorder, they're not scuba diving, they're not mountain climbing, and they can't be stewardesses. They shouldn't be doing anything where there's going to be a pressure change in altitude. Anything else? Good stuff. Okay, so moving on to white blood cells, and the first word I have up here is antibodies, and we all have learned how to type our blood. And the last few of you will be having the opportunity tonight to type your own blood. And you know that involves antibodies. And I've already told you that antibodies are produced by some of your white blood cells. And the job of those white blood cells are to produce the antibodies to protect you from anything foreign that comes into your body. When something foreign comes into your body, those antibodies search it out, attack it, if you will, bind to it, and make a clumping. Now that clumping is a signal now to the rest of your immune system to come and get it, right? So basically when the antibody attaches to the antigen and it clumps, signals are sent out to your immune system saying, I just found something that's not supposed to be here, come get it and destroy it. And that's the wonderful part of our immune system. The problem is, in the meanwhile, when we're waiting for those cells to come and destroy the foreign blood type, we've already caused a small blood clot, right? Not the same kind of blood clot that happens with fibrin, right? But yet still a blockage of blood within your blood vessels. So antibodies, now I'm not talking about all of your immune system here. When I talk about antibodies here, I want you honing in on the idea of blood typing. When and if you take, for example, microbiology, you will get a very beautiful section on how the body fights off bacteria and viruses. And in that discussion, you will learn about the T cells and the B cells and all the different cells of your immune system that are specialized in destroying those foreign invaders. For now, all I'm discussing here really are the antibodies involved with blood clotting, or sorry, with blood typing, okay, with blood typing. But this is a bigger story for a different class with a different emphasis at a different time. But you know so far that antibodies are going to attack foreign blood. So for example, if I have A blood, I would make antibodies against what? B. And if I were to receive that B blood, my immune system and my antibodies to B would search out and find that blood and send the alarm out to my immune system and do everything possible to get rid of that foreign blood. I'm not going to go back through the blood typing story. I hope that is feeling comfortable for you. I know that you have been through that. The last of you are being quizzed on that tonight. You don't want to let this story go, though. If you're struggling with blood typing, make sure you check out with me or Mr. Mueller. Have us go through that with you again. You're going to see it again. You're going to see it on the quiz this week, but you're also going to see it on this next exam, and you're going to see it on the lab practical final. So just know, I hate to see people lose points over and over and over for the same concept. So if you're not making sense of this blood typing thing, search me out, find Mr. Mueller, talk through it. You'll get it. Some people just struggle a little bit longer. To me, it's like riding a bicycle. You skin your knee a couple times, and once you get it, you're not sure why you didn't have it in the beginning. Once you get it, it'll be yours, and it'll make sense forever. Question?
Do you use the same format that Mr. Mueller has?
He uses a slightly different worksheet right, when he describes this. If you'd like, I can certainly go through the way that I do it really quick, if you'd like. There's just different ways of doing this. So let me describe this to you, and I'll make a whiteboard here and go through this. I did the same presentation also on the Lab 8 YouTube video. So if you go under Lab 8, I've been posting all my lab videos, and you'll see a lab link to that. But let's do this. So this is how I approach it. I draw my circle, and I say that is a red blood cell, and let's go ahead and just practice this as if this is an A positive red blood cell. Okay, so the 5 questions I ask you are, number 1, what antigens would be present on the surface of this red blood cell? Let's go back. If you have A positive blood, you received this blood from your parents, right? You genetically inherited this blood type. So what antigens are present on the surface? A and RH. Very good. If you have A and RH, your body would not make antibodies against your own blood, but it would certainly consider what foreign? B. And would therefore, you would be making antibodies against B, anti-B. This could be one of the proteins floating in your blood. Now, because you're making antibodies to B, that means you cannot receive any blood with B in it. Because if you receive B blood, you're going to clot, right? You're going to agglutinate. So I'm going to draw an inward arrow with an X through it. You cannot receive blood from whom? Anybody with B in it. So that would be your B positive and B negative friends. That would also be your AB positive and negative friends. Right? They all have B in it. Can't receive it. If I did, my blood would clump. There are 8 blood types. I can't receive those 4 because they all have B in them, but I can, inward arrow, receive blood from whom? Everybody else. My A friends, plus and minus, and my O friends, plus and minus. Right? They don't have B, so I can receive it, no problem. Now, when I go to give the blood away, outward arrow, what am I giving away? A and RH. That means the people who receive this blood must already be okay with A and RH. So what blood types already have both A and RH in their name? A positive and AB positive. Their immune systems say, hey, I'm cool with A, I'm cool with RH, give it to me.
That's the part that confuses me. What about O positive?
People, think about this. People with O positive would be making antibodies against A. Okay? So go back and listen. If you want to hear this again, go back and listen to this little area. We're about 73 minutes into the lecture. And listen to the 5 questions that I presented and logically answer those questions in the same order. It makes sense. Should I go through one more?
Yes, yes.
B negative. What's on the surface? Just B. What would be foreign? A and RH. So I'm going to make antibodies against A and against RH. 2 different antibodies. Because I'm making antibodies against A and RH, I cannot receive X. I cannot receive anything with A or RH in its name. That includes who? My A friends, my AB friends, because they have A in it, and my O friends and my B positive friends. O positive and B positive friends. Every one of those 6 blood types either has A in it or RH in it. If I received any of those, my blood would clot.
So if you're positive for RH, you can receive from RH, but you just can't give it to somebody.
Okay? So I've eliminated 6 blood types that I cannot receive, but I can receive the other 2. I need to think about question number 4. Question number 4, what's left? What's left? Well, my own blood type and O negative. O negative will always be part of this answer because O negative, remember, is your universal donor. Everyone can receive O negative. Now, when you give your blood away, you said this is where you're struggling. What are you giving away? B. Right? When that person with B negative donates their blood, what are you sending to the recipient? B. That means that the person receiving it, their own immune system must say, I'm cool with B. Well, who's cool with B? People who have it, right? So the only people who can receive B negative blood already have B. Well, that's your B friends, plus and minus, and your AB friends, plus and minus, because they all have B. And they can receive that blood without pissing off their immune system.
Now, if they were positive, could they still receive both?
If who was positive,
If in the circle you have B negative,
right?
if it was B positive,
Because I'm giving away RH,
so you can't give it to
negatives,
the negatives.
right? Just go back and look at the A positive story, right? And the A positive story, you're giving away A and RH. You can only give it to people who already have both A and RH. I think drawing the little circles is helpful. It's just the way my simple little mind works. And there's many, many, many ways to present blood typing. And hopefully, I know a lot of you, and I'll take some heat on this, did not find or could not find the Physio-X exercise on blood typing. Okay? Now, I know that you may not have found it, but I also only received about 3 emails from people saying, I can't find it. Right? So those who found it very helpful. So if you did not have the opportunity to do that little Physio-X 9.1 blood typing exercise, go into Mastering, go to Study Area, and right next to the PAL 3.0 link, there was a Physio 9.1 link. Go into there, go into Physio-X, and there'll be a blood lab and a blood typing lab. And you actually pick up little droppers and add the reagents, the antibodies and the antigens. And I think by going through that process and typing your own blood in lab, you will begin to better understand this process. Have light bulbs shown brighter, or are we still struggling a little bit? Brighter? Sweet, okay? So again, there's dozens of ways of presenting this. This is just the way my mind very logically goes through those 5 questions. Number 1, what antigens? Number 2, what antibodies are produced? If I know what antibodies are produced, I know what I cannot receive. If I know what I cannot receive, I can just blindly put in what I can receive. And then when I give my blood away, I'm thinking about what antigens am I donating, and I can only give it to people who already have that particular combination of antigens. I plead with you, do not try to memorize a table. In a moment of emergency, if you were the one and only person making a decision on a blood transfusion in an emergency, your memory might fail you. Your logic likely won't. So take the 30 extra seconds necessary to logically think through this and get the right answer, rather than, oops, I wasn't thinking, or I just tried to memorize something and I forgot about it. So I encourage you, on the next exam, lecture exam, somebody, case study, is going to definitely be in an accident and is going to need to receive blood. And then they are going to be so thankful that they were able to receive blood in their time of need, they're going to want to now go and become a donor. And you're going to have to answer the same questions. From whom and to whom can you safely receive blood? Okay, so make sure you're comfortable with this story. You're going to see it again, lecture exam, and you're definitely going to see one station of it on the lab exam, where you'll be asked, what's that blood type? Okay? Okay, so that's the whole antibody story. Now, when you go back and look at this table, it may make more sense to you. And remember that if you're O negative, you are considered the universal donor. If you're O negative, the blood bank's calling you every 6 weeks. Please, please come, please come, please come. It only accounts for about 6 to 7% of the population, but it's the blood that we want to have on stock. So you're very popular with the blood bank if you're O negative. If you have AB positive blood, you are the universal acceptor, and you can receive anyone's blood if you have AB positive. And that's also rather rare, about 4% of the population. I don't have those numbers for you to memorize. If you go to redcross.com, you'll see really a nice little unit. I need to incorporate that into my lab and lecture, but like the percentages of Caucasians, African Americans, Latinos, and Asians, and their percentage of blood types across their groups. It's kind of interesting. Okay, so now let's talk about leukocytes a little bit. Again, your white blood cells are there. They are true cells. They have a nucleus. They divide. They have all of the normal characteristics of a living cell. They're bigger than red blood cells. I already mentioned that in that table. About 1.5 to 3 times bigger. And when you see them next to red blood cells, you will see that they're bigger. They don't have hemoglobin. They don't carry gases. That's not their job. Their numbers, again, were in that table. 5 to 10,000 per microliter. Relatively rare. If you had less than 5,000 white blood cells per microliter of blood, you would not have you'd have a lower than normal number of white blood cells. And they would say that you have leukopenia, or you are leukopenic. Osteopenia, a deficiency in bone. Leukopenia, a deficiency in white blood cells. There could be a number of different reasons for that. Or you could have more than expected. So you go in and you've got a spike in your white blood cell count. That means you probably have an active infection and your body is ramping up the number of white blood cells. There are other reasons as well. You would now have leukocytosis. Osis, an abnormal condition of many cytoleucos, right? Many white blood cells, right? Abnormally high white blood cells. A few more facts about leukocytes. There are 5 types, 5 groups, 5 classes of leukocytes. We're going to break them into 2 groups, granulocytes and agranulocytes. Granulocytes have little granular appearances to them. The agranulocytes have the absence of these little granules. Look at this picture. Now, for this semester, Biology 105, you are not expected to recognize these different white blood cells. We will dive in deeper and differentiate these next semester. We'll have a whole blood lab or 2, and we'll talk about all the differences and how to tell these cells one from another. For now, they're just all white blood cells. But 2 groups. On the top, neutrophils, eosinophils, basophils. They are all granulocytes. All that means is if you look at these cells, you're going to be struck by the presence of these little dots, all these little granules within them. Okay? So they are the granulocytes. Now, you can spell these backwards, Ben, B-E-N, or maybe you know somebody named Neb, right? Whatever. Ben has bumps. Ben has acne. Ben has whatever. Ben has the granules, right? So the basophils, the eosinophils, and the neutrophils have the granules within them. I'll say it now, but some of these cells are better at attacking bacteria. Some of these cells are better at attacking parasites. Some of these cells are better at attacking viruses. And when they attack, they release the products of these granules. So they release these products, and that kills the bacteria, that kills the virus, that helps with your immune system. On the bottom are the 2 agranulocytes. This includes your lymphocytes and your